zDNA is commonly believed to provide torsional strain relief during transcription, and it is associated with negative supercoiling. However, while supercoiling is associated with both DNA transcription and replication, zDNA formation is primarily linked to the rate of transcription. A study of human chromosome 22 showed a correlation between zDNA forming regions and promoter regions for nuclear factor I. This suggests that transcription in some human genes may be regulated by zDNA formation and nuclear factor I activation. zDNA sequences downstream of promoter regions have been shown to stimulate transcription. The greatest increase in activity is observed when the zDNA sequence is placed 3 helical turns after the promoter sequence. Furthermore, zDNA is unlikely to form nucleosomes, which are often located after a zDNA forming sequence. Because of this property, zDNA is hypothesized to code for nucleosome positioning. Since the placement of nucleosomes influences the binding of transcription factors, zDNA is thought to regulate the rate of transcription. Developed behind the pathway of RNA polymerase through negative supercoiling, zDNA formed via active transcription has been shown to increase genetic instability, creating a propensity towards mutagenesis near promoters. A study on Escherichia coli found that gene deletions spontaneously occur in plasmid regions containing zDNA forming sequences. In mammalian cells, the presence of such sequences was found to produce large genomic fragment deletions due to chromosomal double-strand breaks. Both of these genetic modifications have been linked to the gene translocations found in cancers such as leukemia and lymphoma, since breakage regions in tumor cells have been plotted around zDNA forming sequences. However, the smaller deletions in bacterial plasmids have been associated with replication slippage, while the larger deletions associated with mammalian cells are caused by non-homologous end joining repair, which is known to be prone to error. The toxic effect of ethidium bromide on trypanosomes is caused by shift of their kinetoplastid DNA to Z form. The shift is caused by intercalation of ETBR and subsequent loosening of DNA structure that leads to unwinding of DNA, shift to Z form, and inhibition of DNA replication.